anyone talks about infection, one of the things that they talk about, or four of the things they talk about, are these particular definitions. So pathogen is the type of infection. It's the microbe. Is it this type of bacteria? Is it this type of virus? What is actually causing the infection? How did that infection get into the body? That's what portal of entry is. So each infection will have a different way. Influenza and COVID-19 are infections that are inhaled through droplets. They're droplet nuclei mode of transmission, whereas C. diff is a contact because you were in contact with infected stool or spores that were present. So it depends on the infection itself how it gains entry into the body. Pathogenicity is describing what the microbe does, what the pathogen does in order to to replicate and take over and cause symptoms. Virulence speaks to the strength of the pathogen. So if a pathogen is very virulent, it does not take a long exposure to get the infection. It could just be a tiny exposure because the hardiness or the strength of that pathogen is so strong. So pathogenicity is sort of the characteristics of the pathogen that help it to divide and take space up in your body, and virulence is the strength of that pathogen. When you're talking about infections, this is what's called the epidemiology triangle, or these factors come together and can end up leading to outbreaks of problems or explain why a certain person got an infection or at increased risk of infection. So there are the agent factors, which we were just talking about. What kind of pathogen is it? What was our Is the pathogen very strong? What are its characteristics? The host factors are things about us that might make us more susceptible or to infection, or might make it harder for an infection to take hold. Our nutritional status of ourselves could be that maybe we're malnourished, then the body is usually weaker and is not as able to fight off infection as if that wasn't a problem of ours, then we wouldn't have that issue. If we were immunocompromised for some reason because we're on chemotherapy or something like that, it's going to be a lot easier to get an infection. An environment is things that can increase the chance that we are in contact with different infections. If we're all living close together, overcrowded, if we are in an area where we're not sure that our water is actually clean, or there's more pollution, so that also affects whether or not people in a certain area will get an infection. So these pathogens can be described in terms of what is causing them. Viruses, bacteria, fungus, and parasites. The order that this is written in also describes how complex the organism is. The virus is just a small piece of genetic material, either DNA or RNA, that's wrapped in a little envelope of protein to get in the body. It's a very simple structure. Bacteria is its own cell, but it is a more simple cell in comparison to ours. So it can live outside of our cells, but it does injure the cells nearby. Whereas fungus and parasites are more complex structures. So it just changes what we need to do in order to treat them. So the biggest class, especially the ones that we have medications for, is bacteria. It's harder to treat viruses because viruses live inside your own cell. So in order to get at a virus, oftentimes that means that the host cell or your cell is also hurt. So it's much harder to treat them. We have a lot more treatment options for bacteria. And so we have a lot of stuff that we do to classify the bacteria to be sure that we are targeting it correctly. So if someone has an infection, one of the things we as nurses need to do is assess that person because we want to know if whatever treatment is given improves the patient's symptoms. So just like when we were talking about congestive heart failure, we focused on the symptoms of congestive heart failure. We also have to focus on the symptoms of different infections, and we would expect that once the patient is starting to have treatment, those symptoms will improve, and when the person is done with treatment, the symptoms should be gone, of, especially for bacteria. So oftentimes people, you kind of know what organisms are present in a certain community, and you don't necessarily have to culture or get a lab results for every infection, but sometimes we do. And so that is the first thing that is done. So that's a picture, that picture of the person holding that petri dish is, that's the culture material. So if a person is, if the provider orders a culture, so this is important, if a culture is ordered, it's best to get this first prior to starting medication. You will get the best culture if you get that material prior to starting medication. So that's desired. Sometimes it doesn't happen that way. The patient had an infection, they were given a treatment that usually works, it wasn't working, so we get the culture later. But usually, if the idea is that we should get a culture, it should be done prior to starting medication. And then if a culture for bacteria is done, there are certain stages, certain things that are done to test the culture, to test the bacteria, to try to name it and discover what type it is. So the first thing that's done is it's stained with a special violet staining to see if it's a gram-negative or a gram-positive bacteria. The gram-positive bacteria stain darker, and it's because of the characteristic of their cell wall. Gram-positive bacteria are easier to treat. Gram-negative bacteria do not take up the stain as brightly because they have very thick cell walls, and that's why they are harder to get rid of. Then we look at the shape and see, is it circular, which is,
You said the gram negative had the thick cell wall?
yes.
Okay. I'm just trying to remember microbiology, so thank you.
No problem. No problem. So, and then we look at the shape. So we see, is it look like circles or rods? Is, are the circles in clusters? Are the rods stuck together? These shapes let us know, kind of help determine what family of bacteria we might be in. So the two that are the most common are bacilli, which are the rods, and coccus, which are the circles. And then they might be in different patterns. These other ones are not as common, but they are also possible and help to name that bacteria. The next part is, so we stained it, we looked at the shape. The next part is to see how, what is the metabolism, or how does this bacteria make energy? We human beings require oxygen to get the best amount of energy possible. And some bacteria are also like that. So that means they're aerobic. They get the most energy when oxygen is present. Anaerobic bacteria do not require oxygen for energy. So that makes them also harder to treat because the host, you, could be very sick and the organism will be just fine. You know, if I have an infection and the infectious agent needs oxygen to live, well, so do I. And so if I am oxygen deprived, all of my cells will be oxygen deprived, including the infection. But if if it isn't, it doesn't need that, and I get really sick, this an anaerobic bacteria could still be dividing because it doesn't need my oxygen to live. So that can make them a little bit harder to treat. And then the next part is, so we you can kind of figure out what an antibiotic, or sorry, what a bacteria is by its gram stain, its shape, how it uses energy, and then so then you get, if you were the nurse in the hospital, the lab might call you and say, okay, yes, this is gram positive bacillus infection, consistent with uh, enterococcus infection. Okay. Um, but now the next part of that is, okay, we've named the agent. How are we going to treat it? And there is a lot of bacterial resistance in the world because of inappropriate use of antibiotics. So this is usually the time, if we were all in class together, I would have you raise your arm and take a pledge that you would never again bully your primary care provider into giving you antibiotics when you actually have a virus, because that is one of the main reasons or main ways that antibiotic resistance occurs. Um, because you have bacteria in your own body, and you can teach the bacteria in your own body by exposing them, by exposing your bacteria to antibiotics, you can teach your bacteria how to evade antibiotics. You can teach your bacteria how to be resistant to antibiotics. And then when you're infected, your bacteria that's present can then teach your infection how to be resistant to antibiotics. So
It's
if I don't take, go ahead.
it's
Does someone have a question?
almost like a game of spy. I'm gonna sneak
Yeah,
in like you, and then I'm gonna learn
it
everything I
is.
need to learn, and then I'm just gonna come in and act like you and test stuff up.
That's basically Lillian, I like your analogies. Yeah, that's exactly what it is. The you're just letting bacteria are very simple. Even our bacteria, because we have tons of bacteria in our body that help our body run, and they're constantly mutating, and they have special um, flagella or almost like an arm where they can kind of connect to a nearby bacteria and give information to the next bacteria. Like, here, I learned this. I'm gonna teach you. So
Isn't
when you
why isn't that why HIV and AIDS are like hard to treat because they're like the masters of that kind of stuff?
Yeah, they do mutate. So once you all viruses have high mutation ability, very high mutation ability. So once you expose a virus to a medication, if you stop taking it, the virus that remains that will end up dividing has already learned how to avoid it. So you can never stop, you can never take a break on your HIV medication, or you'll end up with a resistant HIV. And so the reason why, just because this is the situation we're in, the whole reason why we have to learn this way instead of in the classroom, the reason why we have gotten, this is like the third time that a coronavirus has switched species. We had avian flu, which was another coronavirus. That was the original SARS, H1N1. That occurred when I was working in the ER, but that wasn't as severe for us in Wisconsin. And then we had MERS, which was another mutation of the coronavirus in which it jumped species from camels to people. And that was it was called MERS because it in particular took place in the Middle East. So we didn't have to worry about that because it didn't really travel our way. But now this most recent coronavirus, it hasn't really been completely determined which, because coronaviruses are very common in animals in general, which animal it may potentially have come from, if it was a bat or whatever it is. But what happens is viruses mutate all the time, and if they mutate to switch their surface proteins and are able to connect to a new species, like a person, then you get a new infection. And so this one is a lot better at infecting lots and lots of people than those other two mutations were that we've had in the past. Um, Yeah, so it's in fact that's one of the large arm of infectious disease is always testing animals in Asia and Persia because those it seems to be where those I don't know if it's just because of the preponderance of animals or what the deal is, but that's where sometimes those explosions take place. So that is really important to monitor agriculture because it's oftentimes where viral species jump. So anyway, that's why we're in this damn mess. Um, so if I'm back to what we were talking about, because bacteria have also learned how to be resistant to many of the antibiotics we've used or have used to treat them. We now have to be sure that we're picking the right one, and that's what getting a resistance and sensitivity or test to an antibiotic to a for a bacteria. So that's what this is showing actually. So this whole plate is full of bacteria, like it's nothing but bacteria. These little circles, the middle circle and this one right here, those are full of different types of antibiotics. So we know that an antibiotic worked when around the little white circle there's areas of gray because that killed the bacteria. So that means that these ones that have that like circle of gray around the white, those are representing antibiotics that would actually work against the bacteria. This one is just has a tiny bit, so it's not as good. This one has nothing. That would be a resistant bacteria. So that's how they do that to figure out which antibiotic would work and which probably isn't the best choice. So, and we can't not talk about infections without talking about hospital acquired infections because this is where nursing has a big role. Hospital acquired infections are cost a lot of heartache for patients. It prolongs hospitalization. It also is a big money expenditure for everybody, for the patient who's getting billed, for the hospital. Now hospitals lose money for hospital acquired infections. If the infection happened because of hospitalization, the hospital does not get reimbursed for that. So that's kind of how, in our system, that's how we get change to happen by affecting the bottom line. So the most common hospital acquired infection is a urinary tract infection, and that is in general related to having a Foley. So there are lots of rules around that as a nurse. Um, we should use Foleys for limited time, if at all. If we do use them, then they should be cared for properly with appropriate pericare, not pulling to increase trauma at the area and keeping the area clean. Those are all nursing interventions to prevent a very horrible hospital acquired infection. Um, so I always bring that up. And then we'll stop with this slide before we take our lunch break. But antibiotics then can be described as either broad or narrow spectrum. So some antibiotics, if they're broad spectrum, that means they can kill multiple types of bacteria. And some are narrow, meaning they are only for a select type of bacteria. Most are broad, there's only a couple narrow ones. And antibiotics themselves will have several mechanisms to take care of a bacteria. They will either disrupt cell wall synthesis, so that means they break the cell wall so that the bacteria is destroyed, or they will do something to manipulate the cell's ability to produce proteins or to replicate. So changing folic acid in DNA will change the ability to replicate, and protein synthesis changes the bacteria's ability to care for itself. So some bacteria are what are called bactericidal, and some antibiotics, sorry, are what are called bactericidal, meaning they actually kill the bacteria on contact. And some are bacteriostatic, meaning they stop the ability of the bacteria to divide, and eventually they will just die out then. So we will When we come back, we'll go through common directions that we have to give everybody that is taking an antibiotic and common side effects that we always have to tell people about when they're taking an antibiotic. And these are for all antibiotics in general, and then there'll be certain ones that we'll highlight as we go through different antibiotics. So,
I'm sorry, could you repeat that bacteriostatic and bactericidal definitions real quick one more time?
Sure. So bactericidal means that the antibiotic itself did something to kill the bacteria directly. Oftentimes it's the cell wall synthesis, because it'll actually cut the cell wall and it'll just kill the bacteria. So I kind of think of it as like homicidal, bactericidal.
The
Bacteriostatic,
Marines.
what's that like?
Cyto is like the Marines. They come in and mess it up.
Thank you, Lillian. Every time we need something like that, you just chime in. And then static means you're keeping them static in the same spot. You're stopping them from dividing. So you make it so that they can't replicate their DNA or make proteins in order to keep them functioning. And then eventually they'll just die out. So either way is effective, but that's generally what they do. They either kill the bacteria outright or stop the bacteria from dividing.
Thank you.
All right. You're welcome. So I will see you guys again at 1 o'clock. Have a good lunch.